Bismillahirrahmanirrahim. Assalamu alaikum, dear students. This is Dr. Azam Ali from Department of Biochemistry, FRPMC. Today, the practical that we are going to discuss is detection of unknown carbohydrate in the given sample. So the learning objectives for this practical are that students should be able to describe the principle and significance of different tests that we'll do for the detection of carbohydrates, and perform the test for detection of monosaccharides, disaccharides, and polysaccharides. Now, these are the tests for detection of carbohydrate. These include Molisch's test, Iodine test, Benedict test, Carbohydrate test, and Sullivanov test. This is the general scheme for detection of unknown carbohydrate in a given sample. So this Molisch's test is a general test for detection of carbohydrate, and when this test is positive, afterwards we perform Iodine test, and Iodine test is for the detection of polysaccharides. Now in polysaccharides, in Iodine test, they may give different colors. For example, stars give blue color, dextrin will give purple color, and glycogen will give reddish brown color. And in case if the Iodine test is negative, we move towards Benedict test, which is for the detection of reducing sugars. And we know that there are two groups in reducing sugars. In reducing sugars, these are glucose, fructose, maltose, and lactose. And among the non-reducing sugars is sucrose. So sucrose will give negative Benedict test. Now, once we detect that the given sample contain a reducing sugar, then we further differentiate between reducing monosaccharides and reducing disaccharide, and we do so by the help of Carbohydrate test. Now in Carbohydrate test, monosaccharides, they are stronger reducing agents than the reducing disaccharides. So when the Carbohydrate test is positive, then we perform Sullivanov test, which help us in detection of ketose sugars. So in case the Carbohydrate is positive, which means there is a monosaccharide, and we further want to differentiate between aldo monosaccharide and keto monosaccharide, we perform the Sullivanov test. So this is the general scheme for detection of carbohydrate in a given sample. So we begin with Molisch's test. The purpose of this test is to detect the presence of carbohydrate, whether the given sample contains a carbohydrate or not. And the reagents used in this test are Molisch's reagent and concentrated sulfuric acid. Now it is very important that you must know the purpose and the principle of the test. The principle of Molisch's test is that if in a given sample there is a carbohydrate, if it is a polysaccharide, it will first be converted to disaccharide and then into monosaccharide, and this hydrolysis takes place due to sulfuric acid. Now once the carbohydrate has been hydrolyzed to its monosaccharide unit, then it gets dehydrated by the sulfuric acid and they form furfural or furfural derivatives. Now these furfural derivatives will get condensed with alpha naphthol to give a violet colored complex. Now this violet colored complex that will be produced in the test tube will be our observation, and we have to get inference from this, and that inference will be that the given sample contains a carbohydrate. Now this is the reaction that is taking place in this test. First of all, any carbohydrate that will first be converted into a monosaccharide unit, and under the action of sulfuric acid, it will be dehydrated and will be converted to furfural compounds or their derivatives. Now these furfural derivatives, under the action of alpha naphthol, they will be condensed and give us a colored complex. Now once we have detected that the given sample contain carbohydrate, then we perform the Iodine test in order to exclude whether the sample contains a polysaccharide or not. So for this we perform Iodine test. Now the reagents used in iodine test are original solution and iodine solution. Now this original solution is nothing but the given sample in which we have to detect the presence of carbohydrate. So most of the time you will see either original solution or an OS. Now the principle of iodine test. Iodine test is more of a physical test, but not a chemical test, unlike the Molisch's test. Basically what happens that helically coiled polysaccharides, they adsorb the smaller molecules of iodine on the surface to form a colored complex, and different polysaccharides give different colors with iodine test. For example, starch will give blue color, dextrin will give reddish purple color, and glycogen will give reddish brown color. So you must remember the principle, and an important thing here to note is that this is not a chemical reaction but rather a physical reaction. Now the third test that we perform after the iodine test is Benedict test, and the purpose of doing this test is to detect the presence or absence of reducing sugars. Now what are these reducing sugars? Reducing sugars are sugars with free aldehyde or ketone group. The reagent that we are going to use in this test is Benedict qualitative reagent, which comprises of copper sulfate, sodium carbonate, and sodium citrate. So the principle of Benedict test. In alkaline medium, a reducing sugar, as I told you, reducing sugars are sugars with free aldehyde or ketone group. Now what these reducing sugars do in the presence of an alkaline medium, they reduce the cupric ions to the cuprous ions. Now where did this cuprous ion come from? They come from the copper sulfate, and this alkaline medium is provided by sodium carbonate. So these reducing sugars will reduce the cupric ions to cuprous ions, and simultaneously the sugar will be oxidized in the reaction. And these are the equations which are describing the underlying reaction that is taking place. First of all, sugar in the alkaline medium get converted into anhydride compounds. Now, these anhydride compounds are stronger reducing agents than the sugar itself. So these anhydride compounds will convert, or they reduce the cupric ions to cuprous ions. Now these cuprous ions will combine with hydroxyl group to form cuprous hydroxide. Now when we heat these cuprous hydroxide, they will form cuprous oxide, and these cuprous oxide will give us the red precipitate that we take in the test. Again, a similar reaction. The reducing sugar is first converted to anhydride compounds, and these anhydride compounds will reduce cupric ions to cuprous ions, and when we heat these cuprous ions, they are converted to cuprous oxide. So this is the general scheme for the detection of carbohydrate in the given sample. Now in this table you can see that there are experiment column, observation, and inferences. You don't have to remember all these steps that are included in the experiment column, but rather you have to remember the observation and inferences. So the observation for Molisch's test is appearance of purple-violet ring at the junction of two chemicals, which shows that the given sample contain a carbohydrate. An observation for the iodine test will be either a color change or no color change. If there is a color change, the inference will be polysaccharide present, and if there is no color change, inference will be polysaccharide absent. And in the Benedict test, there will be appearance of red precipitates, and as I told you that these red precipitates are composed of cuprous oxide. So the inference will be reducing sugar present, and if there is no appearance of red precipitate, then the reducing sugar is absent. So in today's session, we will only perform 3 tests with Molisch's test, iodine test, and Benedict test.
So students, we are here at the lab, and now we'll perform all the tests pertaining to detection of carbohydrate. So we'll begin with the Molisch's test. For this we'll take 5 ml of original solution. Original solution is the solution or the sample which contain the unknown carbohydrate which we have to detect. After this we'll add 6 drops of 1% alcoholic alpha naphthol. And now we'll add sulfuric acid. And mind it, this is concentrated sulfuric acid. So we'll add 2 ml of concentrated sulfuric acid along the wall of the test tube. So this was Molisch's test. Now you can see in this test tube that there is a ring formation at the junction of 2 solutions. So formation of this ring is telling us that this test is positive and the given sample contains a carbohydrate in it. Now the second test, which is iodine test. And the purpose of performing this test is detection of polysaccharide in the given sample. I have explained you earlier that iodine is not a chemical test, but it's more of a physical test. So for iodine test, we will take 5 ml of original solution. So here we are taking 2 samples. One of the sample contains a polysaccharide, and the other sample does not contain the polysaccharide. It contains other monosaccharide or disaccharide sugar. So we now add 5 ml of the second sample to another test tube. Now we'll add the iodine solution. Only 2 drops are required. We'll shake the test tubes now. So you can see in the first test tube that we added the iodine, there's a color change, and this blue color is showing that the given sample contains a polysaccharide. And in the other test tube, there is a slight color change, but this is the original color of the iodine. This is not the color change because of presence of polysaccharide, rather it is color change because of iodine solution itself. So the appearance of this blue color is showing that the sample contains a polysaccharide. Now we'll perform the third test, that is Benedict's test. The purpose of performing Benedict test is to differentiate between reducing and non-reducing sugars. The reducing sugars will give a positive Benedict test, while non-reducing sugars will give a negative Benedict test. We'll take 5 ml of Benedict's reagents and to this we'll add 8 drops of original solution. We'll now place this test tube in the water bath and heat it for 2 minutes. We'll boil it for 2 minutes. Okay, after boiling it for 2 minutes in the water bath, now we'll see what changes have appeared. Now this is the observation for Benedict test. You can see in this test tube that red precipitates have appeared in it, which is showing a positive Benedict test, and these red precipitates are formed because of formation of cuprous oxide. Thank you.